Hi, everybody, and welcome to the lecture on the cell, the parts of the cell, and what the different organelles do within the cell. Remember, this PowerPoint is available to you on Canvas. You can use it to take notes on or to follow along, but we're going to be talking about kind of the typical animal cell that you see pictured here on the right side of the screen, with a fairly central nucleus and multiple organelles throughout. Okay, so there isn't really one typical cell in the human body. I told you that form and function are related to one another in cells. So the shape of a cell is going to be related to its function. So the, quote, typical cell that I'm showing you, no, it doesn't really exist per se, but it's got all the parts to it that you will need. And then once you know this basic structure, then you can see how it changes in different cell types. All right, the study of cells is called cytology. I've told you for this root here means cell. Ology is study of. In order to study cells, you have to be able to see them. And so the first way that we ever had to really see cells was light microscopy. And light microscopy works by shining a light through a sample and then magnifying that image through very high quality glass. So it was only when they had high enough quality glass that they could stack those magnifying lenses one on top of the other, which allowed us to finally see that there were individual cells. The structures of the cells are most easily seen if you stain the tissue before you look at it through a light microscope. So light microscopy works by looking at tissues, usually using stains. And this is the sort of image you would see through a light microscope. All right, more recently, we've developed electron microscopes, and there are two different varieties of images you can see with an electron microscope. We have what's called a TEM, or transmission electron microscope, and that is going to allow us to see fine structures within a sample. And then we have SEMs, or a scanning electron microscope, which allows us to see superficial structures or things on the outside. So all three of these images are showing the exact same type of tissue. And we can see here that these cells are tall. We're going to learn these are called columnar. And that on the surface, what's called the apical surface, we have these long projections, which are known as cilia. And you can see those pictured over here on the right. The TEM allows us to see the details on the inside. For example, you can really see the membrane that surrounds the outside of the nucleus. Here, the nucleus is shown in contrast because of the staining. And with an SEM, you can't see the nucleus at all because you're looking at the outside of the cell. Another difference is light microscope, fairly inexpensive. Okay, pretty much anybody can learn how to use these, and access is not limited. These two are much less common and very expensive. So we don't have any electron microscopes at the college for you to use, even if we were there in person, versus we have lots of light microscopes. All right, so the three parts that we're going to be talking about are the plasmalemma, which is the outer, you can think of it like a skin or a structure that surrounds the cell and separates the inside of the cell from everything that's outside of the cell. So it's playing the same role that your skin does. Cytosol, cyte, again, cell, sol is referring to a liquid. So this is the liquid component to the cell. And then the organelles, or little organs, are the structures that you see within the cell. The first part we're going to look at then is the plasmalemma. Now, this is a term that has a lot of synonyms, words that mean the exact same thing. You'll see cell membrane, you'll see plasma membrane, you'll see plasmalemma. They're all talking about the same thing. Now, when we talked about chemistry, I mentioned that among the lipids, there's a very important phospholipid molecule. So if you remember, I said it looks a bit like a sperm with two tails, or you can think about it like a lollipop with two handles. We've got this round head and these two tails that extend outward. Sorry, downward, I should say. And I told you that half of this molecule likes water and half of this molecule hates water. So the heads are known as hydrophilic or water loving. So they can easily come in contact with water. But the tails are hydrophobic. They hate water. And so the tails will not mix with the water, same way oil and water don't mix. And the tails will naturally be drawn to associating with other tails. And this means that even if you have just a whole bunch of random phospholipid molecules and you were to pour them into, say, a cup of water, they would associate themselves in this same configuration you see right here, with the heads being towards the water and the tails being toward one another, okay? So this plasmalemma is made up of a phospholipid, that's what these molecules are, bilayer. Bi means two, Bi. Okay, so it's a double layer of phospholipid molecules. Okay, so it's made up of phospholipid bilayer, and here you see those phospholipids again, pretty recognizable once you know what they look like. Heads love water, tails hate water. And so overall, this membrane that is produced by the phospholipid bilayer is very flexible. I tend to think of it as like a trampoline, or if you're old enough, you might know what a waterbed is, something that offers resistance if you push on it, but it's very flexible. Okay, and it is insoluble in water, meaning it will not break down in water. And that's all because of those tails that hate water. So what keeps it together and keeps it in this particular arrangement with the tails facing the tails, it's just the unique chemical properties of these phospholipid molecules, okay? All right, what does this plasmalemma or skin do for you? First of all, it serves as a site of sensitivity. And what this means is kind of interacting a little bit with the world outside. And this is accomplished by receptors that are embedded in the plasma membrane. And they can recognize and bind with molecules that are outside the cell. So this is going to be important for the cell when it comes to bringing necessary molecules inside. So cell feeding that we'll talk about relies on sensitivity. And these same receptors can be exploited for infection purposes by things like viruses. So those receptors can be used by viruses to recognize what kind of cells they should inject their genetic material into. Okay, and so these receptors also allow communication between cells. We'll talk about that a little bit later on. Okay, the plasmalemma also creates physical isolation. So it separates, as I said, the contents of the cell from everything that's outside the cell, much like your skin does. And finally, the plasmalemma helps to maintain the shape of the cell. It's part of the support system. We'll look at this in a moment, but there are other molecules inside the cell that together with the plasmalemma really give it its shape and creates a structure inside the cell to move materials around inside the cell, almost like kind of an internal scaffolding. All right, and then the last thing is the regulation of exchange. This is controlling which molecules can go in, which ones can go out, which ones can do both. Okay, so that means that a cell is called selectively permeable. It's kind of picky about what comes in and out of the cell, but that ability to be selective is due to the plasma membrane, okay? So why is it important to be selectively permeable? Well, some molecules, if they came inside the cell, might be bad for the cell. They might poison the cell or accumulate to toxic levels. If all molecules could just leave the cell might lose all of its ATP or all of its water, okay? So you want things to be able to come in and out of the cell so it can feed and get rid of waste, but you need to be careful about which materials and how much. Okay, so remember, that's this structure here shown in kind of this grayish blue. Here it's labeled as plasma membrane. So it goes all the way around the cell, and it really allows the cell to interact with the world outside the cell while being separate from the world outside the cell. Okay, now let's look at that transportation. How do materials get in? How do materials get out of a cell? There's two basic ways that can happen. The first set of categories are known as passive. So if something is passive, it does not require energy. And if you've had chemistry, you've talked about diffusion, and you probably understand how that works. But if you're not so sure, diffusion is movement of molecules from an area of high concentration to low concentration. So usually in lecture, I would illustrate this by having you imagine that I'm standing in the front of the room and I drop something on the ground, something really stinky, like a concentrated smell of skunk. And you can imagine how that smell would slowly move from where I dropped it, and it's now in a liquid form on the floor. That smell would extend outward. So the first people to smell that would be the people closest to that liquid, okay? And the reason that those stink molecules are moving is by diffusion. So molecules have energy. You can imagine them kind of shaking or quivering and bumping into all the other molecules around them. And as they do that, as they sort of bang into each other, that allows the more concentrated molecules, like the skunk smell right there, to spread out. So there is no energy required for that skunk smell to travel from the area where I dropped the vial to the people in the first row and then the second row and so forth. Okay, so that's what diffusion is. Now in a cell, small molecules such as oxygen and carbon dioxide are able to diffuse directly across the plasma membrane. This means that because they're so small, they sort of slip between those phospholipid molecules. So if you have a high concentration of oxygen outside a cell, the oxygen molecules will diffuse into the cell, all right? And that's what happens when blood brings oxygen to tissues. And then carbon dioxide is a waste product that accumulates inside the cells due to metabolism. The CO2 levels build up in the cell, and the CO2 will diffuse across the plasma lemma and out of the cells. Okay, so this allows movement without the cell using any energy at all. Okay, there's a special case of diffusion known as osmosis. And osmosis is only used to describe the diffusion of water molecules across a semi-permeable membrane, which I told you, that's how we describe the plasma lemma. Okay, so it's very similar to diffusion as I just explained it, but for biochemical reasons, we treat this separately. I want you to understand though, that if the water concentration is higher outside of a cell, the water will diffuse through osmosis into the cell. And the same will happen if the water concentration inside the cell is greater than outside, then the water will leave, passing through the plasma lemma by osmosis. So think about if any of you have ever been in the hospital or the emergency room, they might have given you an IV. And when they give you an IV, maybe you were nosy and asked, what are you putting into my body? It is not pure water. Okay, they're not just hooking you up to some spring water. They are giving you water that has been mixed with salts. And the reason is they want to put into your veins water at the same concentration as water is already in your blood. Because if they pumped pure 100% water into your veins, then the red blood cells would have all this water moving inside the cells through osmosis. And in fact, your red blood cells would explode. So Ringer's solution is the typical material they use, and it has the same concentration of water as blood does. And that's why this phenomenon of osmosis. All right, and then the last type of passive transport. So there's 3 types of passive transport: diffusion, osmosis, and facilitated diffusion. So facilitated diffusion is the same thing as diffusion. However, these molecules are larger, and they are too large to pass between the phospholipid molecules like the oxygen and the CO2 can. But under certain conditions, larger molecules can move by diffusion if there's an opening for them. And this opening here is called a channel protein. So notice the channel protein, okay, it's a big protein, right? It's made of amino acids. It is embedded in the plasmalemma. So there's my plasmalemma. And it opens both outside the cell and inside the cell. All right, so this means that certain molecules, not all, but certain molecules can move from high to low concentration through this opening. And when that occurs, we call this facilitated diffusion. The cell can control facilitated diffusion because the cell controls how many channel proteins and what kind of channel proteins there are. So if a large molecule that won't fit through this channel tries to move, it can't get through this little tunnel. If too many of the wrong types of molecules are crossing, then the cell can break down this channel protein. If they need more to move through facilitated diffusion, then the cell can construct more of these channel proteins. All right. Now, those 3 were passive, no energy required. And if you remember in the last lecture, I talked about cell energy, and hopefully you remember that is a molecule called ATP. All right, so now we're going to look at active transport mechanisms. And these are ways that materials are moved across the plasmalemma, but they always require ATP, which you see me moving the cursor around. You see this little ATP guy down here, okay? That is the cell's energy. Okay, so let's get oriented. We've got our phospholipid bilayer here. Okay, so we know we're looking at a plasmalemma. Tells you this is extracellular fluid. That means something outside the cell. And cytoplasm, which is referring to the contents of the cell. So this is inside the cell, plasmalemma, outside the cell. All right, so the first type of, excuse me, active transport is an ion pump. If you recall, the ions are charged particles. You can see right here how some of these have little pluses on them. Okay, and then we've also got these guys down here. So the letter K, that stands for potassium, and the Na stands for sodium. Okay, so these have a positive charge because each of them has lost an electron, and these will be moving across the plasmalemma. You will talk about how and why these move a lot in physiology. There's a lot of material on ions in physiology, but for right now, I'm just going to explain the basics of how ions can move. So with an ion pump, the cell uses energy, ATP, to move the ions, but against the concentration gradient. Now, the concentration gradient is referring to where there's a high concentration and a low concentration. Okay, so notice if you look at this sodium here, inside the cell, I see 1 sodium. Outside the cell, I see 3 sodiums. So if we were allowing diffusion and the sodiums were to move from high concentration to low concentration, then they do this, they come inside. But ion pumps allow the cell to go the other way. So now the cell is going to take the sodiums from low concentration and move them to high concentration. That's what we mean by moving against the concentration gradient. So don't worry about what you do once you have this high concentration of sodium ions. You'll get into that later. But what I want you to understand is that an ion pump is an active form of transport. It uses energy and it moves ions from low concentration to high concentration. And if you're still having trouble imagining what that means, I want you to consider, like, if you had a rock and you were standing in the middle of a hill. If you were to put the rock on the ground and step away, it would naturally roll downhill, right? It'd be moving from high altitude to low altitude. Okay, that would be moving with the concentration gradient. And you wouldn't have to put any energy, just stand back and gravity will do the rest. But let's say you put the rock down, but you wanted to go up the hill. If you wanted to go up the hill, you're gonna have to put work in. And that work is ATP. So if you wanna go from low altitude to high altitude, you have to do some work. All right, I hope this helps everybody to understand active transport and ion pumps. All right, the second type of active transport mechanism that we're going to look at is called endocytosis. Endo is a prefix that means within or inside. Cell, okay, so this is within the cell. It moves volumes of material into the cell. These are 3 types of endocytosis. Phagocytosis, which means moving food in the cell. Pinocytosis, which means moving liquid inside the cell. And receptor-mediated endocytosis, which means to bring into the cell a specific molecule. All right, let's look at these. Okay, endocytosis. Here we have a cell. This is cytoplasm, so that's inside the cell. And right here, this dark line, that's the plasmalemma. And I could ask you, for example, what kind of an image is this? Okay, there's no stain, there's no colored stain. This is not like microscopy. We're looking inside the cell. So this is a TEM, or transmission electron micrograph, taken by the microscope. All right, so now we can see inside the cell. So how is this cell going to do this? Well, what it does is it basically folds outward portion of that highly flexible plasmalemma. Okay, this is called making an invagination. Invagination is to form inward, make a pocket, right? A vagina is basically starts growing as a pocket in female development. So that's the idea. So you take the plasmalemma, you fold it inward, and in this case, you surround the object that this cell is going to eat. So we're seeing phagocytosis. The cell is trying to eat this bacteria. So this invagination will form completely around the bacteria so that you have this sphere, okay, like a globe that surrounds the bacteria. And that will move into the cell, right? Endocytosis. Now, in this case, the cell is eating the bacteria, not because it's hungry, but because it wants to destroy the bacteria. This is a way of getting rid of bacteria in your body. So in this case, what happens is the cell has this sphere known as a vesicle. All these little spheres are called vesicles. See the term vesicle here. But this one is loaded with nasty molecules. Okay, it's known as a lysosome. And so these nasty molecules destroy bonds. So you take this vesicle filled with this destructive enzyme and you merge together the plasmalemma. Well, sorry, not plasmalemma. It's it does derive from the plasma element. Phospholipid bilayer is a safer way of saying it. Anyway, you merge these two together because they just they're so flexible, they'll just join right up. That destroys the bacteria. And then the cell is going to get rid of it because it's not trying to eat it, it's just trying to destroy it. So the opposite of this invagination and surrounding is going to happen, where the vesicle now merges with the plasmalemma and dumps the material outside of the cell's body. So if you're getting it out of the cell, that is exocytosis. Here is, like I said, a TEM, and this is showing the same process, but within the vesicles that you've made are not bacteria. So it's not phagocytosis, but liquid. So this is pinocytosis. Let's go back here. So pinocytosis is when the cell eats liquids. Phagocytosis is when the cell eats something else, something more, something solid, okay? So these little spheres are filled with liquid now. All right, so lots of new terminology. A vesicle is a sphere of phospholipid. Okay, it's just a big, it's a way of separating contents from being in direct contact with the cytoplasm. Okay, and so now we've seen endocytosis, exocytosis, and the cell can do endo and exocytosis in eating, drinking, or bringing in particular molecules, the receptor-mediated endocytosis. All right, you might want to take a break now, shake your head. There's a lot of stuff going on here. All right, ready to move on? All right, we are still looking at plasmalemma. We're not done with the plasmalemma yet. Now, over here we have structures called microvilli. Micro means small, villi means fingers. So I want you to think of these as little tiny fingers. And the way the cell makes these little tiny fingers is to fold the plasmalemma, okay? And what this does is it creates a lot more surface area on the cell. Hopefully everybody's familiar with the physical idea of a surface area. But surface area is an important physical property that shows up over and over again in biology. One way of thinking about this is absorbance. Like if you're making a material that's very absorbent, like a sponge, you'll notice that your sponge is filled with tons of little holes. And all those holes have their own surface area of the sponge. And by making a material with lots and lots of surface area, it creates a material that can absorb far more liquid than if you had no holes in your sponge. You wouldn't want to make a solid sponge. All right, so hopefully that helps you to understand. But just imagine if you took your fingers and slowly pushed them into a phospholipid bilayer and then withdrew them and left behind the imprint of all your fingers. So all of this is phospholipid bilayer, and it gives the cell a whole bunch of surface area, which is going to be important in things like absorbing nutrients in digestion. All right, so that is all I want to say right now about the plasmalemma, the plasma membrane. And I told you that the second topic was going to be the liquid component of the cell, and that is the cytosol. But we've also been seeing this term here, cytoplasm. Sometimes people mistakenly think that these two are the same thing. They're really not. The cytoplasm is referring to everything, liquid and organelles, all together. That is cytoplasm. So again, think of it kind of like a math problem. Cytosol plus organelles equals cytoplasm. All right, so everything inside is the cytoplasm, but we're going to start by looking first at the liquid. All right, so the cytosol, this is the fluid, the liquid component, and it consists of dissolved nutrients, okay, and small molecules. So mix it, it's not 100% water, okay, it's mostly water, but it has these dissolved nutrients and small molecules in it. Now, considered to be part of the cytosol are some other types of molecules called inclusions. Inclusions are specifically insoluble. They do not dissolve in water. Okay, fat, we know fat doesn't dissolve in water, and fat is stored inside cells. And another example of an inclusion would be melanin. And melanin is a molecule that gives color to your skin, to your hair. We'll talk about melanin some more later on, but both of these are considered to be part of the cytosol. All right, so that's all I'm going to say. We're not getting into the chemistry of cytosol or anything like that. You will probably get some more of that later in your physiology class. All right, so now that we know we've got this liquid component, let's look at the remainder of the cytoplasm. And those are going to be referring to the organelles. There's two kinds of organelles. There's this first category here, non-membranous. So a non-membranous organelle is not surrounded by phospholipids. Instead, the non-membranous organelles come in direct contact with the cytosol. All right. Now, what are examples of non-membranous organelles? These ones: cytoskeleton, that is the skeleton of the cell. Structures called centrioles, which we are going to talk about when we look at how cells divide. So these guys down here. Flagella and cilia, which are both structures that push a cell along or push materials across cells. In humans, the only cell that has a flagella is a sperm. And the cells with cilia are going to do things like move the egg across the surface as the egg passes through the uterine tube. Right? So these are for movement. And then the last non-membranous organelle is something called a ribosome. And the ribosome is absolutely dead essential to every cell because it's where proteins are made. All right, let's look at a little bit of detail. First, cytoskeleton. This is the internal scaffolding or skeleton of the cell. So I said that the plasma lemma interacts with this internal scaffolding to create the shape of the cell. Over here we see an image of a cell that has been color enhanced using a computer to show you some examples of the cytoskeleton. So this internal support is made, again, of proteins. You should be getting the impression that everything is made of proteins because that's pretty true. And we've got some different examples here. We have microtubule, we have intermediate and microfilament. These are different types of what are called linear proteins. So they're long, skinny proteins, more like string. And you can see that in this particular cell, this area cytoskeleton is largely made up of microtubules. But when you get over to this portion, you're going to see the microfilaments and the intermediate filaments. Okay, so throughout the cell, there's going to be multiple types of proteins making this system that gives strength and flexibility to the cell. So it really acts similar to a skeletal system does for you. All right, next, the centrioles. I mentioned that those are for cell division. Centrioles are also made up of proteins, and this time not a variety of proteins, but this one down here called a microtubule. And a lot of these non-membranous organelles are made out of microtubules. So if it ever comes up on an exam where I ask you what type of protein some non-membranous organelle is made up of, if you have to guess, go with microtubule. That is the most likely answer. All right, now let's look at these structures involved in movement. So here on the bottom, we see a flagellated sperm. Okay, sometimes there are ones that have more than two tails, but that is not normal or typical. Okay, so these cells should have just one long tail-like structure, and this is called a flagellum. Just so you know, because a lot of these words are Latin or Greek based, sometimes it's a little difficult to recognize the singular versus the plural. The term flagellum means one of these. Flagella means more than one. Okay, it's a little odd. All right, then the cilia, there's multiple areas where we find cilia in the human body. Here's one example. The cilia are on the surface, what's called the apical surface. They're on the apical surface of these particular columnar epithelial cells that we'll be learning about in histology. If you look at an SEM, it shows you what the cilia look like. To me, they look kind of like seagrass. If you've ever been in the ocean and you look down and you see those long, thin green grasses that sort of move back and forth in the waves. Or sometimes I imagine them like a bunch of arms extended upward in a mosh pit, if you're more into that kind of thing. But I would just want you to imagine long, thin structures that extend upward from a cell, and these move. So in this case, this diagram shows you what the cells look like that line your trachea. Okay, remember your trachea, if you haven't looked at any of that stuff in a while, the trachea is where the gas passes going to and from your lungs, right? So when you breathe, the air comes in your trachea, and the trachea is lined with mucus, this very sticky material. So if you have been breathing polluted air, which you live in Southern California, so I hate to tell you, you are breathing in polluted air, the dirt that you inhale can get stuck in this mucus. All right, then the cilia, which are constantly moving, move in the direction of carrying the mucus away from the lungs, up the trachea. Okay, so this mucus will eventually go up to the top of the trachea and reach a point where the next time you swallow, mucus will be pushed down into your stomach and digested. The point is that the cilia and the mucus prevent particles of dirt or pollen or other things that we inhale, prevent them from going into your lungs. It's a protective mechanism because lungs are very delicate. Another way to think about this is if you clear your throat, you go like that, you are pushing the mucus upward. So if you inhale something irritating, you might find yourself coughing. That's the same idea. But the cilia do this all the time and help to keep the lungs clean. Now, the last example is a very, very common non-membranous organelle. They fill your cells. You have tons of these guys called ribosomes. They come in two little pieces. They fit together. If you took Bio 1, you spent some time looking at ribosomes and how they play a role in the process called translation, which is how the cell builds proteins. I'm not going to ask you to go over the details of translation or transcription for this class, but you'll probably run across it again at some point. The ribosomes may either be stuck to other structures, and if they are stuck on other structures, they're called fixed. Okay, so here where we see an TEM, look at these little dots, these little dark stain or these dark colored dots. Each one of these dots is a ribosome, and it is stuck to a different type of organelle called a rough endoplasmic reticulum. So all these little dots here, these are all fixed ribosomes. And then the free ribosomes are just floating around in the cytosol. Okay, so two places you can find ribosomes are either fixed or free. All right, now we're going to go on to the organelles that are membranous. That means that the organelle is surrounded by a phospholipid bilayer, which keeps this organelle separate from the cytosol. So it means that different functions can be carried out in these organelles without involving the rest of the cell. This organelle is quite recognizable. Okay, it looks maybe a little bit like a caterpillar, or not sure what you see when you look at them, but they are fairly narrow and longer. They have kind of a larval-like quality to me. They look sort of like it's going to develop into a bug or something. This is a mitochondria. And mitochondria, you can think of them as the powerhouses for the cell. They're like a nuclear power plant or a coal burning plant because they convert energy from one form to another. So in this case, the mitochondria converts energy from, say, the handful of almonds you ate 20 minutes ago, and they turn it into ATP. Okay, so the more of these mitochondria you have in a cell, the more energy that cell is going to be using. So you can, again, relate form and function. So the function of that cell is related to the fact that it needs a whole bunch of mitochondria. If you see fewer mitochondria, then this is a cell that is not using up as much ATP and doesn't need as much ATP. All right, now everybody can probably recognize this central structure in a cell that's called the nucleus. It's shown here perfectly round. It's not obviously perfectly round, but when we look at cells, the nucleus will often appear round. Sometimes it looks oval. So again, the outside here, this is a phospholipid bilayer. But inside the nucleus is the DNA. So each cell has usually one nucleus. We'll talk about some weird cells that have more than one nucleus. But this nucleus is filled with DNA. And I told you that DNA contains the directions for how to make proteins, right? Okay, so it's like a cookbook that tells the cell how to make every possible protein that the cell could ever want. Another thing found within the nucleus, and you might find more than one, is called nucleoli. So this is the plural. It's one nucleolus, two nucleoli. Okay, so these nucleoli, here's one in it right now, are tiny spheres, circles, within this big nucleus. And their job is to make ribosomes. Okay, remember we need lots and lots of ribosomes. So that's what the nucleolus or nucleoli will do. All right, so the nucleus toward the center of the cell usually, surrounded by a phospholipid bilayer, contains DNA, contains the nucleoli. And there are ways of moving between the nucleus and outside the nucleus where the cytosol and other organelles are. There are basically openings. Okay, so the nuclear envelope is very much like the plasma lemma. It is selectively permeable. Some things can go in and out of the nucleus and some things cannot. For example, the DNA cannot leave the nucleus. All right, so they are, the nucleus is also selectively permeable. All right, now there are two different types of membranous organelles with the category endoplasmic reticulum. There's the rough variety and the smooth variety. And these names have nothing to do with function. It's just when people were first looking at cells through early microscopes, they saw things and they just based the names on what they looked like. So this was just, they just saw membranes inside a cell and some were bumpy and some were not bumpy. So they said, okay, that one's rough, that one's smooth. And if you look in the cell, the two are continuous with one another. Okay, so on the left, we have the rough endoplasmic reticulum. On the right, we have the smooth. So you would think that they carry out very similar functions, but actually they don't. Over here on the left, the rough endoplasmic reticulum is covered with ribosomes. And I told you these put together the proteins. They attach one amino acid to another amino acid. But actually, the process, when the proteins leave the ribosomes, the proteins are not in their correct shape. So the proteins that have been strung together will go inside this rough endoplasmic reticulum, and they'll be modified or changed so that they're in the right folding pattern. Because the shapes of the proteins are very complicated, and the proteins will only work if they have the correct shape. So that will take place inside rough endoplasmic reticulum. So it's part of this process of making proteins. All right, and so the proteins will pass from the ribosomes into the rough endoplasmic reticulum. But over here on the right-hand side where you don't see any ribosomes, this is smooth endoplasmic reticulum. So abbreviation ER means endoplasmic reticulum, but don't assume you can abbreviate on tests, okay? I want full answers. Once you pass the class, then you can call it ER if you want, but first you got to prove that you know it's called endoplasmic reticulum. Its job is twofold. One is to make lipids or fats, and then the second job is to break down toxins. So organs in your body that break down toxins, the big one is the liver. Okay, anytime you eat anything, it's going to be passing through the liver. And so if you eat something that is toxic or drink something that is toxic, that will be broken down so that you don't poison yourself, hopefully. And to break down toxins, you need lots and lots of smooth endoplasmic reticulum. And so you're going to find lots of cells loaded down with that particular type of organelle if you look in the liver. All right, the next membranous organelle is called a Golgi apparatus. And to me, when you look at them in cross-section, it reminds me of a stack of plates, like in your cupboard where you got plates and then you got some bowls and cups. Just imagine them sort of stacked one on top of another. Okay, and they're found close to rough endoplasmic reticulum, where we are folding the proteins. All right, so we have our folded proteins. They're all ready to go, and they're inside the rough endoplasmic reticulum. Now, what do you do with those proteins? Some part of the cell has got to figure out where those proteins should go. Kind of like a FedEx center, if you think about shipping and handling, that's what's going on in the Golgi apparatus. So, little pinched off phospholipid balls, remember these are called vesicles, are going to carry the completed proteins over to and then merge directly with. See how they just merge together? They're going to merge right into the Golgi apparatus. And then the Golgi apparatus can determine what to do with them. There's three things that can happen to these newly produced proteins. One, they can become part of the plasma membrane. Okay, so they just come right up here and the vesicle just merges right in together with the rest of the plasmalemma. And now inside this, there'll be a whole bunch of useful proteins that are going to be used by the cell. Maybe it's going to be a channel protein. Maybe it's going to be a protein for communicating. Okay, so there's lots of different proteins in the surface of the cell. Second possibility for a newly made protein is that maybe it's going to be released to the world outside the cell, dumped out of the cell. Okay, and we'll talk later about this, but let's imagine, for example, this is a digestive enzyme made by this cell, and now that enzyme is going to be used to break down your lunch. Okay, so you have to get the enzyme out of the cell, and that's the same thing we've already looked at called exocytosis. So in this case, the vesicle is going to merge here and dump. It's almost like spewing its contents, like a little volcano. It's just spitting it all out, okay? And then the last thing is these proteins, some of them are going to be kept inside the cell. So here's a vesicle, and we saw this term before, lysosomes. And lysosomes, anytime you see the word lice, it's not like the little animals that get in your hair. This is LYS. This is referring to breaking things down. So if something lyses, it breaks down stuff. Okay, so this lysosome is filled with nasty little proteins that can break bonds. So you might use this to kill a bacteria, for example. And so those lysosomes are just going to float around inside the cytosol until they're needed to break something down. Here's an example of another type of vesicle that can remain within the cell. So just like the lysosomes can float around until you need to, say, digest a bacteria, the peroxisomes are filled with proteins. Here they are. And these are used not to kill bacteria, but to break down toxins. So you might find lots of these in those liver cells that help to detoxify the food that you've absorbed. All right. So that's a lot of material we've covered. Okay, we're not quite done yet. So you can, remember, you can pause this, you can shake the cobwebs out of your brain, get yourself a cup of coffee, whatever you need. The last thing that I want to talk about, I think it's the last thing, is what's called the cell cycle. And this is really looking at kind of the lifespan of a cell and how cells divide to make more cells. All right, so the cell cycle is represented here on the bottom left. And what we see in pink here, this first 1, 2, 3, 4 stages, this is known as mitosis or cell division. Following cell division, we're going to talk about cytokinesis, which is how you physically separate the two cells. But over here in blue is the other part of this cell's life called interphase. All right, and during interphase, a cell is preparing for mitosis, for cell division. But that said, not every single cell is going to undergo mitosis. Some cells will enter interphase and never leave. They are never going to divide at all. Other cells move very quickly through interphase in order to divide. So examples of this would be like the neurons in your brain. You've probably heard, don't destroy the neurons in your brain because you're not going to get any more. So the neurons in your brain are not undergoing mitosis. You have a finite amount of these neurons. On the other hand, we see very rapid growth and mitosis in cells that line your mouth. So if you've ever, accidentally bit the inside of your cheek, and even if it draws blood and it really hurts, it'll heal up pretty quickly. In just a few days, you won't feel anything at all. So that ability to rapidly repair tissues relies on cells undergoing mitosis quickly. So it really depends on the cell, but all cells are going to experience interphase. Some cells will divide and separate, okay? Now the cells we're talking about here are called somatic cells. A somatic cell is a cell that has the full complement of 46 chromosomes, okay? Or at least that's the typical number in most human beings. So these are all the cells of your body, with the exception of the cells that make egg and sperm. Those are sex cells, and they're going to undergo a different process called meiosis. Okay, so we're just focusing on somatic cells. So these could be in your brain, these could be lining the cheek, anything like that. All right, so let's look at this blue part first, interphase. So what can happen during interphase? Well, cells will grow, they have to copy their DNA, and they have to grow some more. And all of these have to happen if a cell is going to divide. If a cell is never going to divide, you can just stay right there, and you pretty much don't even need to grow. You can just pause right there in G1. So the way to think about this is, imagine if you were going to undergo division, if you could replicate yourself asexually, and there are organisms that can. But right now, let's say you're, 5'6" and 150 pounds. Okay, in order to make two individuals identical to how you are now, you would have to have, instead of one heart, you need two. Instead of 2 lungs, you'd need 4. You have to duplicate everything in your body. You'd also have to get twice as large. So if you're going to make 2 little copies of yourself, you need to weigh 300 pounds to replace that 150-pound person twice. So think about it in that perspective. So the cell has to get bigger, it has to synthesize, and the most important thing to copy is the DNA. The DNA, remember, that's in the nucleus, and it has the information on how to make proteins. So you have to copy this, and you have to copy it perfectly. Because if it's not perfect, the next cell won't be able to make proteins and it might die. Okay, so all of that is the cell cycle. This can take a very short period of time. This can take an entire lifespan of a cell. It really depends. Now, this idea of synthesis. How do you perfectly copy DNA? Well, it all relies on these four guys down here, these words: adenine, guanine, cytosine, thymine. Okay, these are the four different types of bases that we can find in DNA. The shape of DNA, if you look over here on the left side, you've probably heard this before, but imagine if you took just an old-fashioned ladder. Okay, a ladder has the side rails, and then it has the things you step on, right? These are the steps, these are the rails. And you took that ladder and then you twisted it. Okay, that's what this is called, a double helix. So all you're doing is taking a ladder and twisting it. So let's look at the steps. Each step has two parts and they're color-coded. So notice this one in red, that's adenine. This one in blue is thymine. What that tells us is whenever you have an A or an adenine on one side, you'll have a T or thymine on the other. These two pair up together. And in the same way, the remaining two bases will also pair up together. And that's guanine and cytosine. So to make exact copies, all the cell does is break open right through the steps this double helix structure. So imagine you just cut that ladder right in half. And now you have half a step. But that half a step tells the cell what other half should be its complement. So the complement is the missing portion. Over here, this is one half the original DNA, and now the cell is building the missing half of DNA as a complement to the half it had. So anywhere there's a blue, you know there's a red missing. Anywhere there's a green, you know it must be a purple. If it's red, this must be blue. If it's purple, this must be green. So basically, one half of the molecule is all you need to tell the cell how to build the missing half. That's what we mean by saying that the strands of DNA are complementary to each other. All right, so the cell in that synthesis stage of interphase splits the DNA molecules, uses each half to build two complete copies that are now identical to one another. That is called DNA replication. All right, now, for the stages of mitosis, there are four stages. They're called prophase, metaphase, anaphase, and telophase. All right, and they're pretty complicated, and I've just kind of boiled it down to the simplest things. And I want you to understand these steps, and I want you to be able to identify at which phase these occur. Okay, so we're going to start off with prophase. Now with prophase, let's look over here at a typical cell. Okay, and here's light microscopy. All right, so the outside, there's my plasma membrane. The inside, this is my nucleus. Okay, the first thing we can talk about is the nuclear envelope. The outside of the nucleus disappears. Not poof in a magic sense, but the cell breaks down all the pieces that it uses to build the nuclear envelope. So all the molecules are still there. They're just all split apart. All right. Inside the nucleus, while this envelope was disappearing and breaking down, the DNA started becoming more condensed. It started sort of wrapping up on itself. And the result is that you get these really obvious structures called chromosomes. Now, in the daily life of a cell, the DNA is all stretched out, all extended within the nucleus, so that the cell can easily find the recipe it needs for each and every protein. But during cell division, the chromosomes appear and the DNA becomes very, very dense. So each of these little X's that you see is one chromosome, right? So all of this, we're looking at prophase. All right. The next phase is called metaphase. Meta means middle. So those chromosomes that appeared in prophase are going to line up. You can see them down here. They're all lined up at what we call the equator. So that's the midline. If you treat the cell like the Earth, like a globe, it has one pole here, and then an equator in the middle. So in metaphase, the chromosomes line up at the equator. And remember the centrioles we talked about? I said they were involved in cell division. Well, the centrioles go to either pole. So we have centrioles up here at the North Pole and centrioles down here at the South Pole. And their job is to split up the chromosomes. They do that by producing these rope-like structures called spindle fibers. So these spindle fibers, again, they're not actually ropes. These are made of proteins. These are extending from the centrioles to attach to the chromosomes. Okay, that's metaphase. Next phase here, anaphase. And what happens in anaphase is these spindle fibers contract to separate that X shape of a chromosome into two separate parts. So you pull the two parts separate. So here, notice how they're an X and they're held together in the middle. That middle portion breaks. So instead of having the letter X, it's going to look like two L's, right? It's just two straight structures. And those two halves are being pulled to opposite poles by the spindle fibers. That's anaphase. And then in telophase, we just reverse everything we did in prophase. So the DNA is going to unravel and the chromosomes will no longer be visible. And the nuclear envelopes reform. But now we have two, one for each cell. Okay, that is the end of mitosis. But this is still one big cell. You have all the fixings for two cells, but they're not physically separated. The separation into two cells comes after, sort of overlapping with mitosis, but it is not part of mitosis, and that is called cytokinesis. Think of this as cell cutting. This is when the cell separates. Okay, so let's see if I can show you. Let's see if I can get this to come up. Okay, here's my video, right? And then I have to make sure I'm sharing that with you. What are we doing here? Let's stop that. Okay, so now I just have to make sure. Come here. Sorry, I just want to make sure it's still sharing. Where's Zoom? There it is. Okay. This is the hardest part about lecturing this way, is just not messing up the technology. Okay. So if I am, okay, you should be able to see this now. This is a YouTube video, and we're looking inside a cell. There's some mitochondria, Golgi apparatus. There's the nucleus. We're going to go into the nucleus. And what we're seeing at the end of interphase is that the chromosomes are going to start to condense. Here they are. There goes the nuclear envelope. So we're in prophase. There's centrioles. Okay, and the chromosomes are lining up at the midline. So that's metaphase. See the spindle fibers from each side? They're going to pull these two parts. Okay, there they go. The two pieces are separating. That is anaphase. And now that's telophase. We've got 2 nuclei now. Each one's got DNA inside it. And the last thing that happens is cytokinesis. Watch it, pinches in, separates into 2 cells. Okay, that's cytokinesis. All right. And I believe I've got a second video to show you. There are tons of videos online, and I would absolutely encourage you to take a look at as many as you can. Let's see, maybe we just have to copy it. Oh, that's the same one. That didn't help me. Okay. Use this one. Here we go. Okay. And I'm still screen sharing, so you should be able to see this. Making chromosomes. So we're inside the nucleus. So notice the centrioles are moving to the poles. Spindle fibers are coming out. Okay, so we're still in prophase. Okay, remember the spindle fibers are coming from the centrioles. They're lining up on the midline. This is metaphase. Pulling, anaphase. See how they're separating? Two nuclei now. On the outside, this is cytokinesis. See how it pinches in? All right, so hopefully those 2 videos helped you to understand what I was trying to speak about, but please feel free to look. There are tons of different images on YouTube. And if you have any questions, just feel free to ask me during office hours if you're having any trouble with these. All right. There we go. All right, so let's finish up finally. This is a long, long lecture. All right, you need to know the 4 stages. You need to, if I tell you something like chromosomes line up at the equator, you'd have to be able to identify that that is metaphase. You need to know the order in which they happen. I have a little mnemonic device, so it's PMAT, right? Prophase, metaphase, anaphase, and telophase. I don't know why, but I've always remembered that as pretty much anything. PMAT. But come up with your own. Mnemonic devices are best if they're silly or funny or even a little naughty, as long as you can remember them. All right. So I mentioned that some of the proteins in the plasmalemma enable communication and allow cells to basically interact with other cells. Well, in some areas of the body, cells establish very, very tight connections between the individual cells. And this is really important in the cells that line your body in order to help keep out invasive materials like viruses, fungi, bacteria. So your skin, the lining of your mouth, all of that is made by cells called epithelial cells. And epithelial cells are kept quite close together. So how do they do that? Well, the first way to do that is to form what's called a tight junction. And that is formed between proteins that are found in the plasmalemma or cell membrane. And they really hold the surfaces close together. So if you, whoops, come back here. Okay, if you look closely here, a tight junction, you're going to see right here. So notice these little dots. Each one of these is a protein, and it bonds to a protein in the cell next to it. It would kind of be like you holding hands with someone. If you're holding hands with someone, then it would be very awkward for me to walk between the two of you. I'd have to break your grip. So the tight junctions force materials to go directly through the cell if possible. But I told you the cell is selectively permeable. So in this case, a fungus couldn't get between the cells because of the tight junctions, but it couldn't get into the cell either because of the semi-permeable membrane. And that helps to protect the deeper tissues from any kind of an infection. The second is what's known as an anchoring junction, which is again a way of holding two cells close together. But this is really strong because the proteins that connect are actually working through the cytoskeleton. So remember the cytoskeleton, those are the proteins inside the cell. So if we look closely at an example of an anchoring junction, we can see that the connection is made including the cytoskeleton. So this would be kind of like, instead of just holding hands with somebody, when you're holding hands, your bones grow together, right? Think how strong that is. I couldn't push that apart easily if you had had your bones grow into the bones of the person next to you, right? So that's really strong. That's an anchoring junction. And then last are communicating junctions, which are well named because they allow communication. So how does this happen? Well, let's look over here. This is an example of a type of communicating junction called a gap junction. And in blue, we have channel proteins. And we've seen channel proteins because they let little molecules move across a plasma lemma. But in this case, if you link together two channel proteins, that means that now materials from one cell can go all the way into the next cell without ever having to cross a plasma membrane. So going back to the idea of you holding hands, this would be like you're holding hands with somebody and your blood flows into their blood. Okay, so you have this direct link from person to person. So in this case, it's not so much about strength as it is about molecular communication. So molecules from one cell can move right into the next cell, okay? So these structures here are known as connexons, and it allows communication by small molecules. All right, I just gave you an enormous amount of material to make up for the short lecture I also gave you this week. So please watch it carefully, listen to it again if you need to, write down your questions, and I hope to see you in office hours or answer your questions by email. All right, take care. Bye-bye.